It's a great pleasure to introduce today's seminar speaker, Francine Ke. So some of you might have gathered, I enjoy having a bit of fun before I introduce the students. So today I decided not to poke fun on Francine, because Daniel and I, we decided that she was way too easy a target. And we call her, Daniel and I call her the princess because of her impeccable behavior and also because of her incredible wardrobe. Of course, next to my wardrobe, any wardrobe is quite amazing. But well, also I don't want to poke fun at her because I'm pretty glad I actually managed to get her to come up here and give a talk, because she was the second most nervous student to get to coach to give a seminar. But we're all good now. And I'll give a prize to the person who can name who was my student who was the most nervous one. And a hint, it's about as diametrically opposed in shape, demeanor, and anything else than to Francine that you could imagine. So I'll give a prize afterwards. The second thing I want to say is that yesterday, Francine and I had discussed her talk for the last time, and then it ended up into this really fantastic, almost heated debate about interpretation of the data and what the bigger picture of everything is. And it was really quite tiring for me, maybe even more tiring for her. So afterwards, I needed a long run to recover. And during that long run, it dawned on me, I thought, actually, it's been achieved what the ideal goal is of a PhD. She has actually become to the point to understand all her data better than me and to actually challenge authority in a very major way. For that, I can take only very limited credit, a little bit. And I'd like to publicly thank here especially Philippe, who has been a fantastic co-supervisor for Francine, and even more so, I really want to give a big thank you to Anna Voss. She has done a tremendous job, as you will see from Francine's talk, about teaching both her and me about how to analyze embryos, how to interpret data. So big thank you, Anna, for that. And no, Liam, don't smirk. I don't have anything going with Anna. I actually have something going with Tim. So on that note, Francine will give a talk today about her work on BOC. And this has really been a fantastic and frustrating project at the same time, because while it's an analysis of one specific gene, her work really informs what, at least to me, and I think Anna would agree also to her, is probably some of the biggest questions in the field of cell death, developmental biology, cell biology, full stop. And that is, while we all in the textbooks write and in the grant applications, cell death shapes morphogenesis during development, if you look at the evidence for that, it's actually very poor, and we can't put any names onto genes. We can actually say very little about it. And while Francine's work hasn't answered all these questions, it's actually gone a very, very long way to make us ask even deeper questions on this. So, all to you.
Thanks, Andreas, for the introduction. So, good afternoon, everyone. I'm here. Sorry, I have to fix this mic first. I'm here to share with you some of the work that I've done searching for the physiological role of Bcl-2 family member BOC. So to begin my talk, I'd just like to start with a brief introduction of the intrinsic and extrinsic apoptotic pathway. So in the extrinsic pathway, it's initiated by the ligation of these death receptors that contain an intracellular death domain. And following the ligation of these receptors, it leads to the activation of caspase 8 and subsequent activation of downstream caspases and apoptosis. In the intrinsic pathway of apoptosis, this pathway is regulated by the Bcl-2 family members, and it's initiated by developmental cues, cytotoxic insults, and starts with the binding of BH3-only proteins to pro-survival members such as Bcl-2, and then allowing BAX and BAC to subsequently permeabilize the mitochondrial membrane, leading to the release of cytochrome C, which then leads to, again, activation of caspases and apoptosis. So to tell you more about the Bcl-2 family of proteins, it can be divided into two groups: the pro-survivals, consisting of these members, as well as the pro-apoptotic members. And the pro-apoptotic group can be further divided into two subgroups: the multi-BH domain proteins, consisting of BAX, BAC, and BOC, highlighted in pink, as well as the BH3 only members. So the protein that I'm interested in today, as you know, is BOC, and even though BAX and BAC have been widely researched on, not much is known at all about what BOC does. So when we do an amino acid sequence comparison of BOC with the other members of the Bcl-2 family, we see that it shows the highest sequence similarity to both BAX and BAC. So this is very normal then, that one might assume that for a long time people think BOC must play a similar role like BAX and BAC. So to make things just a bit more complicated, it seems like in the Bcl-2 family, proteins with opposite functions, such as BAC and Bcl-XL, and which also have moderate sequence similarities, can have a similar structural fold. So when you superimpose these two proteins, they seem to look quite alike. So in this instance, it's really hard to predict what BOC does just based on its amino acid sequence or structure, which is still currently unsolved. So as I mentioned before, BAX and BAC have been extensively studied. So, and these mice lacking BAC alone or BAX alone have been generated. So for BAC deficient mice, they are mostly normal, and the cell survival assay showed that there's no statistical difference in the survival of BAC knockout cells compared to wild-type controls. The males and females are fertile, and in the brain, there's discrete accumulation of cells in the periventricular region, and also they display increase in platelet counts. And BAX deficient mice as well are outwardly normal. The spleen although their spleen and lymph nodes are slightly enlarged, and they also show this discrete accumulation of cells in the periventricular region of the brain. However, males are infertile. So we see that when either gene is missing, these mice display only minor abnormalities. However, when both genes are knocked out together, we see a much more dramatic effect. So less than 10% of BAC double knockout mice actually die, most of them die perinatally, and less than 10% survive into adulthood. And they display these dramatic phenotypes. For instance, interdigital webbing in their feet and paws. In the brain of the BAC double knockout mouse, you see accumulation of these cells here that's only in a smaller amount in a single knockout. And they have enlarged spleen and lymph nodes. And the lymphocytes were resistant to a broad range of apoptotic stimuli. So interestingly, despite these profound abnormalities, some of the organs that are thought to be shaped by cell death still develop normally in these animals. For instance, the heart, the lungs, the liver, and the kidneys. So these observations raise some very interesting questions. So what could possibly be functioning to compensate for the absence of these two proteins? So could BOC be responsible for these developmentally programmed cell deaths that still occur? So to answer this question, our lab generated BOC knockout mice, BOC-BAC double knockout mice, and BOC-BAX double knockout mice to see what of the functions of BOC that are unique and those that overlap with BAX and BAC, or BAC. And finally, we also generated BOC-BAC-BAX triple knockout mice. So I'll first start off with introducing what BOC does. And I noticed that a lot of you are laughing because they sound very similar, BOC, BAX, BAC, but just try to bear with me. So what do we know about BOC? It's not much. We don't know much about this protein. It's first isolated from a rat ovarian cDNA library in a yeast-2 hybrid screen for proteins that interact with MCL1. It's highly expressed in reproductive tissues, and it's postulated to be a regulator of germ cell apoptosis during maturation, or it may promote cell death in hormone-dependent cell populations. The BOC promoter region contains HIF1 binding sites, so it may be regulated by hypoxia in certain organs. So some splice variants of BOC have been identified, such as BOC-S here, which is the fusion of the BH3 and the BH1 domain. This splice variant does not heterodimerize with pro-survival proteins and exhibits increased pro-apoptotic activity. Another isoform of BOC, BOC-P, which is identified in humans so far only, is implicated in trophoblast cell death triggered by hypoxia. So a recent study showed that the overexpression of BOC is unable to kill cells lacking both BACs and BAC. This suggests that perhaps BOC may act upstream of these two proteins to induce cell death. And as for the localization of BOC, there has been some findings. So this group mentioned that BOC is localized to the nucleus, while recent findings show that perhaps it's mainly localized on the ER and Golgi membranes, and perhaps in a lesser amount in the mitochondria. So to start off with doing the project, we decided to first look to see where BOC is most abundantly expressed in a healthy animal. So we took a range of organs and subjected them to qPCR analysis and Western blot analysis. And we see here that, as documented, BOC is most highly expressed in the uterus and the ovaries, and also quite high in the brain. And unfortunately, there is no commercial BOC antibody available. So to do this Western blot, we're really lucky that we had some collaborators who were very generous to share with us their homemade polyclonal antibody, which works well because it manages to pick up BOC at the correct size of 23.5 kilodalton. And these Western blot results correlates well with our qPCR data, and the BOC antibody does not react with BOC knockout tissue. So I was also very lucky that when I started my PhD, the BOC knockout mouse had already been made. Just to briefly share with you how it was made. So this is the BOC. So the BOC gene has 5 exons, and the start codon, the ATG start codon is in exon 2. So a targeting construct was made that had LOXP sites inserted between exon 1 and flanking exon 2. So after recombination, the FLOX allele contains this targeting cassette now, and when they were crossed to Cre deleter females, this region is removed, and now we have the deleted BOC gene. So we tested by qPCR analysis, Western blotting, and genotyping, and we saw that in all these cases, there was no more BOC expression and protein. So now we could move on to examine the BOC knockout mouse. So it's actually, it looks quite normal, like similar to the BAC knockout or the BAC's knockout, and the BOC knockout males and females are fertile in contrast to the BAC's knockout males, and they're healthy and normal. So we decided now to look at the organs where BOC is most highly expressed to see if we can find any defects. So I'll just briefly show you the ovary here, and I want to explain this diagram so you can better understand my slides now and later on. So females have a number of primordial follicles. There are immature follicles that surround an immature. There are follicles that surround an immature oocyte. These follicles then mature to become primary, secondary, and antral follicles, which then, during ovulation, the oocyte is being expelled. So what we did was we looked at the histology of the ovaries of BOC knockout and wild-type mice at different ages, at PN11, 14 weeks old, 7 months old, and 12 months old. And we see that there is no difference between the wild-type and BOC knockout ovaries. So we also collaborated with Professor Jeff Ke from Monash University, and what he did was, so we performed serial sections of the whole ovary, and we looked and we examined all the ovary sections and counted the number of follicles in the middle portion of the ovary to see to enumerate the number of follicles that are present in wild-type and BOC knockout ovaries. And as you can see here, however, we noticed that in young mice and old mice, we do not see a difference in the different follicle populations. So we decided next to look at the brain, because that's another organ where BOC is highly expressed. So we collaborated with Anne to do these experiments. So if you recall, the back-spec double knockout mice has an accumulation of these cells here, and so does the single knockout animals. However, when we perform similar analysis by serially sectioning Bok knockout brains, we see that there's no accumulation of such cells. Also, another publication by this group mentioned that Beck's knockout mice had a 50% increase in facial neurons. So we similarly did the same thing, and we counted the facial neurons in wild-type and Bok knockout mice. But unfortunately, we saw that they were also similar, and there was no difference. Then we subsequently moved on to look at the hematopoietic cell subset composition. And as you can see here, so these are FACS analyses. In the thymus, the spleen, the bone marrow, and the blood, there is no difference in leukocyte subsets in these organs. And to examine the mice further, we moved on to look at the other major organs in the Bok knockout, and we see that there is no difference in comparison to wild-type organs. We then decided to look at whether or not the loss of Bok confers a protective effect to lymphocytes when we challenge them with some cytotoxic insults. So we FACS sorted B cells, CD4 and CD8, and double positive thymocytes, macrophages, and neutrophils from the bone marrow, the lymph nodes, and we exposed these cells to a wide range of cytotoxic stimuli. But unfortunately, we also saw that there was no difference at all between the survival of wild-type and Bok knockout lymphocytes in response to these drugs. This made us conclude that the loss of Bok does not have a significant impact on the development and apoptosis in healthy mice. So in 2010, this paper came out mentioning that the genomic region containing the Bok gene is deleted in many cancers. So these authors examined more than 3,000 cancer profiles from 26 different cancer types, and they looked for trends where regions of the gene were amplified or deleted. So here we see, for example, that MCL1 is one of the regions. MCL1 is a gene in one of the regions that's constantly amplified, and Bok is here, and it's constantly deleted. So the question then is asked whether or not Bok can function as a tumor suppressor. So to do this, we used a tumor model that was readily available in our lab, which is the eMimic transgenic mouse model. And however, when we compare the survival curves of eMimic Bok head and eMimic Bok knockout survival curves, we do not see a difference, so suggesting that the loss of Bok does not accelerate eMimic-induced lymphoma development. This is similarly noticed for the loss of Beck, which also does not accelerate eMimic-induced lymphomagenesis, which is in contrast to Beck's, which does. So these results may not be surprising because Bok is not highly expressed in B lymphoid cells. So we wish to now examine other tumor models, especially cancers of tissues where Bok is highly expressed, such as the reproductive organs or the brain. So to conclude the first part of my talk, we found that Bok is expressed in a wide range of tissues, especially in the brain and reproductive organs. Bok-deficient mice are normal, and their lymphoid and myeloid cells respond normally to apoptotic stimuli. And lastly, the loss of Bok does not accelerate lymphoma development in eMimic transgenic mice. So next, we wanted to look at an organ where Bok is highly expressed, for example, the brain, and we wanted to ask the question, what is the role of Bok in stress responses in this organ? So I was very privileged to be able to perform these set of experiments with Professor Jochen Pren at the Royal College of Surgeons in Ireland. So this is a picture of the Giant's Causeway in Northern Ireland. It's one of a World Heritage Site, and it's really pretty. And it's not true that in Ireland it's always raining. They do have nice weather as well. So one of the techniques that I learned in Professor Jochen Pren's lab was to culture organotypic hippocampal slices. So briefly what we do is we take a mouse that's 4 days old, remove the brain, and perform microdissection under the microscope to remove the hippocampus. The hippocampus is then placed under the tissue chopper, which chops up into little pieces, and these pieces are then separated and plated onto these membranes that are sitting in media. So these slices have to be cultured for a week before they are ready to be treated. So subsequently, we expose these slices to either oxygen glucose deprivation or kainate treatment. So So this is how a slice looks like under the microscope. This is the CA1 region of the hippocampus, the CA3 region, and the dentate gyrus. So when we do OGD, we basically remove the media from the slices and put in fresh media that does not contain glucose. And these slices are then put into a hypoxic chamber with about 1 to 2% oxygen content at 37 degrees for 45 minutes. Subsequently, the slices are removed from the chamber and it's placed back onto conditioned media and then into a normal incubator with normal oxygen content. And then after that, the slices are stained with PI to identify the dying cells, and these images are taken. So sham-treated slices are slices that have always been in normal conditions and are only imaged after 24 hours. So as you can see here, at 4 hours, not much death has occurred in the wild-type and Bok knockout slices. But at 8 hours, we do see some cell death occurring in the CA1 region, and this is more prominently observed in Bok knockout slices. And at 24 hours post-OGD, we see that there is even more cell death, and I've graphed these results here. So this is what we see from 5 mice, and at 24 hours, we still see a slight trend that perhaps Bok knockout slices may have more cell death. However, it's possible that at such a late time point where all the cells are dying, it's hard to identify a more significant difference. So with kainate treatment, we did the same thing. Slices were cultured and treated with different concentrations of kainate. So kainate is an agonist for the kainate receptor, which is a ligand-gated ion channel. So in response to ligand binding, the channel opens, allowing the flow of ions. But overstimulation of the receptor can lead to excitotoxicity due to a high influx of calcium ions into the postsynaptic neuron. Then this causes neuronal damage and cell death. So in this context, however, Bok seems to act in a different way. So we see that there's less damage in the CA1, CA3, and dentate gyrus regions in Bok knockout slices compared to wild type. And one of the things that I got to observe when I was in, one of the other things that I got to observe was live mice surgeries. So in vivo middle cerebral artery occlusion is a very commonly used model to mimic stroke. So this is the equipment that we set up before the experiment is conducted. So this equipment here monitors the cerebral blood flow to the brain before and after the occlusion, where you should see an approximate 80% drop. And here you have to perform the surgery using the microscope. This is a probe that measures the animal temperature, and we put the animal to sleep. We apply ointment on its eyes to protect the eyes, and an incision is made around the ears where you stick the probe in to monitor blood flow. Following that, the mice is flipped over, and if you see all these hair-like structures, these are actually monofilaments that are used to occlude the artery. So it's inserted into the common carotid artery and into this internal carotid artery. And so after that, the mouse is closed back up and allowed to recover. And subsequently, this occlusion is removed, and we sacrifice the animal and use the brain for analysis. So the brains are serially sectioned and then stained with cresol violet. And so the healthy tissue stains purple while the infarct area remains unstained. And unfortunately, after a significant amount of work, we noticed that brains from Bok knockout and wild-type mice showed no significant differences in infarct size. So in summary, in kainate-mediated cell death, the loss of Bok provides minor protection in the CA1, CA3, and dentate gyrus regions of hippocampus. And in OGD-mediated cell death, the absence of Bok leads to slightly more cell death when compared to wild-type controls. So perhaps this indicates that depending on the stress stimulus, Bok may have a pro-apoptotic function or a pro-survival function. So we then wanted to see whether or not Bok has an overlapping function with BAX or BAC, seeing that a dramatic effect was only seen when BAX was knocked out together with BAC. So to do this, we generated Bok-BAC and Bok-BAC's double knockout mice. So firstly, We wanted to see, so these are just single knockout matings that were set up, and these are the matings to give us BOC-BAC or BOC-BAC double knockout mice. And we wanted to see whether or not these animals are present at the expected frequency at weaning. So for instance, here, BOC heterozygous mice were being crossed, and we should see 25% of BOC wild-type offspring, 50% of BOC heterozygous offspring, and 25% of BOC knockout offspring. And in this case, for example, the matings were set up such that they were knockout for BOC and heterozygous for BAC. So we should technically get 25% of BOC knockout BAC wild-type mice, about 50% of BOC knockout BAC het mice, and about 25% of double knockout animals. So in this instance, we do get the expected frequency at weaning. We then moved on to examine these mice, and surprisingly, they look physically normal and healthy. With the exception of BAC knockout males, which I mentioned earlier, are sterile. This phenotype is also observed in the BOC-BAC double knockout mice. We then proceeded on to analyze these animals by lymphoid organ analysis by FACS, looking at their thymus, spleen, lymph nodes, and bone marrow. We also did histology and cell survival assays. So I'm just showing you here an example of thymic analysis in young adult mice between 8 to 12 weeks old. And I've also separated my data into males and females. So what you're seeing here is actually thymic analysis of females. So interestingly, we noticed that BOC knockout and BAC knockout thymuses are slightly heavier than the wild-type ones, but this is not seen in the double knockouts. So we're not completely sure why this is the case. However, because the difference is rather minor, we have not thoroughly found out why. We haven't thoroughly done tests to find out the reason. And when we did FACS analyses on the different subsets of T cells in the thymus, we see, so I'm just showing you CD4 positive and CD8 positive thymocyte numbers here. We see that there's no difference between the double knockouts in comparison to single knockouts or wild-type controls. So a similar analysis is carried out in aged mice. So these graphs represent data from 1-year-old females. And we see now that we no longer see this slight increase in BOC knockout and BAC knockout thymic weights. But interestingly, we notice a decrease in BOC-BAC double knockout thymus weights. And when we looked for the different T cell populations, we notice here that perhaps BOC-BAC double knockout mice may show an increase in CD4 positive T cells compared to wild type. But unfortunately, because these results were not consistent with males, so males do not show this, and there's no real obvious phenotype that jumps out, we decided not to pursue these small differences. And similar kinds of results were also obtained in the spleen, and the bone marrow, and the blood. And when we performed cell survival assays, we found that, so once again, cells were FACS sorted from the lymphoid organs and exposed to different stimuli. And I'm just showing you an example of two here, which are B cells and double positive T cells. We see that, so if you notice the legend here, wild type in black and single knockouts in blue, yellow, and pink. And the double knockouts, so BOC-BAC double knockout in green and BOC-BAC double knockout in purple. We notice that there is no difference. So the loss of BOC or BAC alone does not give a protective effect. And even in the combined absence of BOC together with BAC, we do not see any protection. In some cases, for instance, B cells treated with ABIT and etoposide, we see that the loss of BACs alone is sufficient to give a protection, but the loss of BOC on top of BACs does not offer any additive protective effect. We then finally moved on to look at the histology of the other organs in the double knockout mice because we wanted to examine whether or not there were any abnormalities in these organs. So I'm not going to show you all of them, just a few. So here are the testes of mice from testes from different genotypes. And as I mentioned earlier, BAC knockout males are sterile. So you can see here that in all other genotypes where there is mature sperm. We see that in Beck's knockouts, there's an absence of this. Instead, we see a lot of cell death and multinucleated giant cells in the Beck's knockout mice, as well as the Beck's double knockout mice. When we looked at the retina, we noticed, so if you look here, this is the neuron layer of the retina, this one. We see that I've counted these layers, and there are about 5 to 7 layers thick in the wild type Beck knockout, and double knockout animals, Beck double knockout. But however, if you notice the Beck's knockout neuron layer, they are thicker compared to the rest. And this is also seen in the Beck's double knockout. But this phenotype is there is no additive layers in the Beck's double knockout compared to the Beck's knockout mice. And lastly, the other major organs from these animals also appear normal, and I'm just showing you here the kidney, for example. And because Bok is highly expressed in the ovaries, we then went on to see whether or not there were any defects in these double knockout animals. So, however, we noticed that in young ovaries, there were no difference in primordial, primary, or secondary follicle numbers between the different genotypes. However, at the antral follicle stage, we noticed that Beck's knockout females have a decrease in antral follicles compared to wild-type controls. And even though the other genotypes may seem to have a decrease as well, these results were not significant. So when we repeated this analysis in 1-year-old aged females, we do see a difference here. We noticed that the Beck's double knockout mice has much more primordial follicles compared to wild-type Beck knockout and Beck's knockout controls. So this increase is also seen in, although not significant, but there's a trend in the other follicles, in follicles at the other stages. And this may suggest that the combined loss of Beck together with Beck's causes an increase in follicles. So to summarize this part, in most of the organs that we examined, the loss of Beck does not exacerbate the defects caused by loss of Beck or Beck, but the combined loss of Beck and Beck's may result in the prolonged survival of follicles in aged females. So for the last part of my talk, which is also the most exciting part, so you should probably wake up now if you're sleeping, does the loss of Beck together with the absence of Beck's and Beck have an effect on apoptosis and development? So we wanted to examine the combined loss of these 3 proteins using a triple knockout mice. And also we wanted to generate our own Beck's double knockout mouse line to look at the effects of the loss of Beck's and Beck on embryonic development. So here we set up matings, these matings to give us double knockout mice, Beck's double knockout, and these matings to give us triple knockout mice. So the way that they're set up is that they were knockout for Beck, wild type for Beck, and heterozygous for Beck's. And in here, they're knockout for Beck and Beck, and heterozygous for Beck's. These matings have been going on for 10 months and 20 months respectively, and most of the matings gave offspring. However, the amount of survivors were not really high. And when we looked at the time of cell death of these offspring, we saw that most of them were dead at birth or die between day 1 to day 0. So just to mention that for data obtained here, it matches what Linston et al. Saw, that most of the mice, that less than 10% survive into adulthood. So we then went on to see whether or not at weaning, the Beck's double knockout animals and the triple knockout mice are present at the expected Mendelian frequency. And so the way the matings are set up, so for instance here, we should be getting about 25% of wild type Beck knockout Beck's wild type mice, about 50% of wild type Beck knockout Beck's heterozygote mice, and 25% of double knockouts. But we're not seeing this. And for matings to give us triple knockout mice, we saw that we were also losing a lot of these important genotypes at weaning. So we decided then to go back further and go back further into the development and see at which stage we're losing these animals. So I started to collect embryos at E19 just before they were born. And as we can see, for matings that were set up to give us back double knockout mice, we still got the expected ratio that we wanted, so about 25% of double knockouts. But in contrast, in the triple knockout mice, we're already losing these triple knockout embryos at E19. So this suggests to us that BOC could play a critical role in embryonic development that overlaps with the roles of BACs and BAC. So the following data now that I'm going to show you are just some preliminary results, but I think you might be interested to see how these double knockout and triple knockout mice look like. So here's a normal BAC knockout embryo at E19, and all of these are BAC double knockout embryos. And if you notice here, this one has spina bifida and this one has exencephaly, whereas these three, which are also BAC double knockouts, look rather fat and healthy. So this tells us that there are some inconsistencies in the phenotypes of these animals, and not all of them look like this. So as for the triple knockout mice, so these are just BAC double knockout controls that are normal looking, and these embryos are just E14 stage of development. But we see here in the triple knockout mice, this one has a weird cone head and 7 fingers, and these ones have a cleft lip, which is more severe in this one. So again, not all of them look like that, only some of them do. So it suggests that perhaps BOC once again may play a critical role in embryonic development that overlaps with the roles of BACs and BAC. And to finish off, I just want to show you, because as you know, I did manage to collect some of these double knockout and triple knockout mice for analysis, and this is how they looked like. So they were quite runty and sick when I had to take them, and most of them had to be taken due to malocclusion or hydrocephalus. Okay, so we looked at the retina from these animals. If you recall this neuron layer that I was telling you about, which is more thick in the BAC knockout animals. And we see that this thickness is increased further in the BAC double knockout mice, and so as well as these nerve fibers. And however, in the triple knockout, you see that this neuron layer and the nerve fibers are even more increased. So this suggests that perhaps the combined loss of BOC together with BACs and BAC may increase the accumulation of cells in the retina to a greater extent than the loss of BACs alone or in combination with BAC. And when we looked at the various organs in these triple knockout and double knockout mice, so this control represents wild-type single knockout controls that pretty much look normal, as you have saw from my previous slides, and they are around 8 to 12 weeks old. The 5 double knockout mice that I have gotten to analyze so far had to be taken around 4 to 10 weeks old, and they show some infiltrates in the kidney and the liver. And after 20 months of breeding, I only had 2 triple knockout mice to analyze. And as you can see here, the kidney and liver also has significant amounts of infiltrates. So the lymphoid organs of these animals are also structurally disorganized. If you look at the lymph nodes, there's no germinal centers. The thymus has an uneven ratio of cortex to medulla, and in the spleen as well, there's a loss of organization of red pulp and white pulp. So as I mentioned, I don't have many mice to look at, so these data are just preliminary because the animals are too few right now to make any conclusive statements. So to end this part of my talk, we noticed that the triple knockout mice are not present at the expected frequency before birth, while the BAC double knockout mice are. So this may suggest that triple knockout mice are dying early on in development and may have more severe developmental defects than the BAC double knockout mice. So perhaps BOC may act like in a BAC-like manner during embryonic development. Also, it seems that many tissues still are able to develop even in the combined absence of BACs, BAC, and BOC. So this tells us that other processes, in addition to regulated apoptosis, plays a role in morphogenesis. Does Bok play a role in mouse development? Probably not, because we've examined the Bok knockout mouse, and it's largely normal. And I'll leave you here with some interesting questions that arise from our findings. So could Bok, together with Baxenbach and the extrinsic apoptotic pathways, have overlapping critical roles in animal development? Could non-apoptotic cell death processes such as necroptosis play important roles in animal development by themselves or together with Bok? And lastly, are apoptosis and other cell death processes not as critical as we thought they are for the morphogenesis of diverse tissues during animal development? So that's the end. And I'd like to thank all these people. So in MGC, my supervisor Andreas and Philippe and Jerry. So without their support, I probably wouldn't be able to do my experiments and be normal. And also all these other people that helped me. So Lorraine helped me with histology and analyzing my mice when I was away. Michelle helped with taking ovaries from really young mice. Phil was teaching me some stuff when I just joined WEHI. Gemma and Steffi were doing bone marrow reconstitutions for me while I was away, even though I did not show these results yet. Daniel for helpful suggestions, and everyone else in the Strasser Lab and MGC. My friends, Marco, Anne, Alex, Liz, Rima, Sylvia, Anna, Anne, and Tanya, and Dana. So it's really nice to be in MGC because of all these people. In molecular medicine, Anna Voss, which has been super, super supportive and encouraging. And I think without her, I'll have gone crazy. And the WEHI histology department for processing all my slides. Fax facility genotyping. Bruno and Carly for doing the massive amount of genotyping that we have. Jason for blood analysis, my mouse technicians, Jess, Sophie, Stephanie O'Connor, and Giovanni. Our collaborators in Bern, Thomas Kaufman, who made the Bok knockout mouse and also gave me some of his homemade supply of antibody. In Monash University, Jeff, who was helping me look at the ovaries. At the Royal College of Surgeons, Professor Jochen Pren and Helena Bonner, she was the postdoc that was mainly teaching me all these techniques. My scholarship, I'm funded by the Leukemia Foundation Australia, and my travel fellowship to Ireland. And of course, my family and friends, without them, which I couldn't have done this. And so just to show you guys some photos that I've collected throughout my PhD years, that's us when we went for paintball. Andreas was really happy because he's trained to shoot people and stuff, like he's really trained. And also here we had lab outing at the boathouse. This was us when I first joined WEHI, our first year of student retreat. Andreas and his mustache. Us again at the boathouse. This is Daniel trying to hold a pint of beer, I don't know why, for as long as possible. And my friends, thank you so much for your time.
Okay, plenty of time for questions. Devo, and then Ruth.
When Bok was overexpressed that he wasn't able to kill. Do you know why it's not able to kill? Is it something about the structure? Is it, or does it have some function during development that isn't cell death related?
Well, okay, we don't know the structure of Bok, firstly, so we can't predict whether or not it has a pro-survival or apoptotic function. And you're talking about how Bok was not able to kill Baxenbach deficient cells, right? So there was just some very recent work that's still unpublished, where Bok overexpression couldn't kill these cells, but could kill cells that has Baxenbach. So that's why we had the conclusion maybe it might act upstream of these two apoptotic proteins. But with regards to how it works, we honestly do not know. As you can see that my results, some of them show, like only one of them showed a slight protection when we had, more cell death when Bok was removed, and the others show protection. So I think, I don't really know how it does this.
There's a striking difference between Rackenbachs versus the proselyters, is that they change shape and they oligomerize. And those people who have an antibody, perhaps they've looked at that. Do you know if Bok, when cells die, does Bok undergo a shape change or become oligorized using sort of standard techniques? Would you need an antibody or a reactive protein? Perhaps someone's done that?
I think Thomas may have speculated that BOC does undergo dimerization when apoptosis is induced. But are you trying to, do you want to know, can you repeat your question? So it's just, that's what Thomas has, like, may have found because he has this antibody, but otherwise, because we don't know, we have no structural information on BOC or whatsoever, we don't actually know whether or not it undergoes a clear structural change.
So what would be the next double knockout you do with BOC?
I think results show that BOC may most likely be pro-apoptotic. So there are all these questions that I posed at the end of my seminar. So let's say maybe we want to investigate the role of BOC in necroptosis. We could create BOC knockout with 3 knockout lines. Or if we are suddenly interested to find out whether or not BOC may have a pro-survival role, we could knock out BOC together with maybe Bcl-1 or Bcl-2 to see whether or not we see a difference in these mutant animals.
Next was Paul, and then Ross.
And so I was just wondering, with the persistence of that layer of neurons in the triple knockouts, with the brain abnormality and the failure of, for the cranium to close over, with, is it possible that there's some kind of neuronal migration abnormality? And what about other nervous systems? The enteric nervous system, for example, does that form normally?
Well, Paul, to be honest, I haven't looked at that yet. We're starting only to look now at these embryos because what me and Anna discussed to do was that we first wanted to look at the stage where the triple knockouts have gone missing. So if you notice, like at E19, the backspec double knockouts were still present at normal frequencies, but not the triple knockouts. So we have to sort of go back in development and see at which stage these animals start to die off. And only then we can start to investigate which part doesn't form properly, and that would be the best, So we're still collecting more embryos and we need more N numbers.
Ross, and then Murray there.
So Francine, that 2010 paper you mentioned about the human copying over loss in cancers, that's pretty compelling evidence for BOC as a human tumour suppressor. Could you elaborate on what tumour types they were in human and what you're doing in mouse, which non-hematopoietic tumour?
Yeah. So when I read that paper, all the tumors that they analyzed was in the supplementary information. But unfortunately, when I clicked on the supplementary information, it was like a huge Excel sheet of, I don't know, like 1 million pages. But very sadly, they did not clearly document which tumors are each of the genes lost from. So I don't know myself, and I'm very interested to find out. But I guess the first place to look, if we want to find out whether or not BOC plays a role as a tumor suppressor, would be to look in tissues where it's most highly expressed, and also maybe try out some solid tumor models.
That wasn't my question.
Julie.
Given that BOC is so highly expressed in the ovary relative to other tissues, have you looked at the next generation and what happens to the fertility of the BOC knockout mice?
Oh, so you mean when I made BOC knockout mice together and what happens to the ovaries? So we've done lots of extensive histology on these ovaries, and I've even gotten Jeff to look at them. Unfortunately, we don't see any difference between BOC knockout and wild-type ovaries. So,
But is there any change in the fecundity of the animals? Like the number of offspring that they have per litter, or
no.
what about, are they able to have pregnancies at a later age? Because you've speculated that that might be the case because you saw more follicles.
Well, so technically, BOC knockout follicles, yeah, maybe. I have not set up I have not used older mice, older females for matings, because we always try to use the younger females that come through. But that could be a good suggestion, and I could try to do some experiments with BOC knockout females and BOC double knockout females to see whether or not they are more fertile when they're older compared to the other genotypes.
What were you arguing with Andreas about?
Whether BOC has a pro-apoptotic function or a pro-survival function. It was quite heated.
Yeah. What do you think?
What do I think?
Yeah.
Well, because I'm super open, I think, maybe too open to any possibilities. Like, with the work I've done, obviously, it seems that BOC may lean more towards having a pro-apoptotic role. However, I was also very excited to find that maybe it could play a pro-survival role, and we could do experiments to test that. So I was just basically saying to Andreas that we have to be more open, and he's like, we are very open, but... so, yeah, that's about it, but...
If you're going to pay the mouse bills, you're going to be more open. So on this note, it remains to thank Francine for a great talk. And I mean, if anybody, as Philippe says, wants to do quadruple and quintuple knockout mice, they should talk to Francine and me afterward. And I think it's a great lesson that even if you don't have phenomenal phenotypes, you can actually give a quite phenomenal talk, producing a lot of insight. So thank you very much, Francine.
Thank you. What?